Okay, good evening, everyone. Thank you for joining us for our first Zoom after the Chagim to renew and reinvigorate what we have been doing since the beginning of COVID with our opportunity to, as you see on the screen, rise together and make sure that our community continues to be together in every sense during this very, very difficult time in our history. This has been really very uplifting to get together with so many of you over and over again for various educational opportunities. And we do want to, as we have for each of our Zooms, recognize that we continue to hear about and to know so many people who are suffering with COVID, and we pray for their health and their well-being, wishing them all a refuah shlema. And for those, and there are so many within our community and the greater community who have lost loved ones, we extend our condolences and remind you that we are with you during this very, very difficult time period. Please stay strong and know that we are all together and we are going to rise together. And tonight, backed by very popular demand, we have what we have been promising you, an evening with Dr. Sarah Schlesinger Hirschfeld to discuss vaccines and antibody therapy, looking forward on what's to come. And we, over all of the various Zooms that Dr. Schlesinger has done, said to you that this was coming and people have been asking, and so tonight it's here. Dr. Schlesinger, as I'll remind you, is the Associate Professor of Clinical Investigations and a Senior Attending Physician at Rockefeller University. She is best known to us as the mother of Jacob Hirschfeld, who's in 4th grade. And really, we continue to thank Dr. Hirschfeld, Dr. Schlesinger Hirschfeld, for the work that she does, not only on these Zooms, but for the community at large, both for the KJ and Ramaz communities in guiding us through so much of the difficult information that we need to have, and she's so helpful to all of us. As our moderator this evening, we are very privileged to have Lenore Brachot. Lenore received her bachelor's in engineering from Cooper Union and then her master's in engineering from Columbia. And since that time, she has actually been a master teacher in Ramaz and has taken on over these past few years the chairmanship of the science department. She does much, much more than that in our high school. She is a very, very beloved teacher and colleague to so many of us. We're very privileged and thankful that she has agreed to join us this evening to moderate. She is also very well known in the school for her amazing children, Nadav, who's in 4th grade, and Amit and Liana, who are also in the elementary school and in the early childhood. Thank you so much, Lenore. Thank you, Sarah. A reminder that tonight, what we will continue to have is an opportunity after Lenore and Dr. Schlesinger speak. We will have the opportunity for a question and answers. Those questions and answers can be placed in the Q&A at the bottom of your screen. We ask that you remember that Dr. Schlesinger is not going to address personal questions or questions having to do with decision-making for communal issues. Instead, those kinds of questions can be sent privately to either Lenore Brachot or Sarah Schlesinger, and we'll be happy to share their emails with you. But please, let's keep tonight in a very general way. And we thank you both again, and thank you everyone for joining us. Please take it away, Lenore.
Thank you so much, Kenny, for inviting me tonight and for such a nice introduction. I'm honored to be moderating the discussion tonight with Dr. Sarah Schlesinger Hirschfeld, so I promise to call Sarah for the rest of the evening. Sarah has given her time over the past few years to speak in the upper school in different forums. She has spoken to after-school clubs, to grade-wide assemblies, as a guest speaker in class about her research, about being a woman in medicine, about ethics in her field. And we are so appreciative of her giving her time and her knowledge. Just this past Monday, she spoke to a senior elective class and answered all their COVID questions, and it could have went on for hours. So I really appreciate all that you do for, like Kenny does, for the school and for the community. And I'm looking forward to moderating the discussion tonight. And you wanted to begin with talking about immune therapy.
Right. Well, I would just like to say thank you both. And, you know, I realize this is the sort of usual love fest that we get to have. But when Jacob entered Ramaz, he entered in first grade. It was not a usual entry year. And so it was a little bit awkward. And I met Lenore because she invited him sight unseen to a birthday party for Nadav. And she was so kind to me and so kind to Jacob that every time I see her, I just think of that kindness and that warmth and that welcome. And, you know, we've become dear friends and colleagues because ultimately I'm a teacher. That's really what I do. And so it's my great privilege to be able to teach sometimes at Ramaz. When my older boys were at Charles E. Smith Jewish Day School, I taught there. And it's really been a wonderful, enriching part of what I do. So I'm delighted to do it. I wish, frankly, that my area of expertise weren't so interesting right now, but here we are, and it is. And as I told Kenny, I'm so excited because as we've been talking, you know, over since May through the pandemic, I keep saying, we'll talk about vaccines when we have a whole evening to talk about them, because I am what is referred to as a vaccinologist. I have worked since longer than I'd like to admit, but certainly since the early 90s on developing vaccines and immunotherapy, primarily for HIV, sometimes for cancer and for some other rare diseases. But the principles of this apply to what we're experiencing now with COVID. And the first thing that I wanted to discuss is that we are, you know, I am ever optimistic. I am an optimistic person by nature. My mother raised me that way, though I'm not sure it was my nature to start with. She made sure that I learned that pretty quick. And I am every day grateful for that. We are looking at some of the fastest, most effective, and, you know, well done discoveries to treat any viral disease that have ever happened. We've only known about COVID since January, really. So for 10 months. And in that 10 months, this explosion of scientific information and medical progress has been like nothing else anybody's ever seen. It's ever happened. So that's really good. And we're on the precipice of really seeing that come to fruition. Just today, I was watching the news as I was making dinner. Remdesivir, which is a small molecule, a drug that was developed originally for hepatitis C and Ebola, and works quite well for COVID, has been approved by the FDA. It was under an emergency authorization until today. And the immune therapy that we're going to discuss, and most importantly, the vaccines, are truly on the horizon in a way that they're months away. But months right now is a pretty long time because we are firmly in the beginning of what's called the second wave. So from the beginning, we've all understood that it was very likely when fall came and the temperature dropped and people are inside more, that there would be a second wave of COVID. And much as we hoped that wouldn't happen, and much as we've worked and we're still working to suppress it, it appears that that's what's happening now. So the next 6 to 12 weeks are going to be really hard on everybody. And even though we all have COVID fatigue, including me, we are still without a vaccine. We have some drug therapy, but we don't have treatment that can easily be deployed. And so we're going to have to continue to do the hard work that we've been doing to protect ourselves, our families, and our community. I'm going to say a little bit more about that later. But whenever I talk about COVID to any venue, and Lenore will tell you, when I spoke to the high school students who were asking really smart, interesting, esoteric questions, I ended up talking about hand washing at the end, which I'm sure nobody wanted to hear, but it's what we have before us right now. But what's on the horizon is really, really inspiring to me and is comforting as well. And I want to talk about vaccines, and I'm being very careful to watch the time because I also want to get to your questions, because I think I already see a lot of really good questions in the chat. But I want to talk about a little bit before that about the difference between the convalescent plasma treatment, what some people are now referring to as the Regeneron, which is a monoclonal antibody treatment, and a vaccine. Because all three are methods of using the immune system to fight the virus and are used for other viruses as well, and bacteria and fungi and all sorts of things. But they're very confusing even to those of us who spend our days working with them. So the first principle, the first methodology, which is called convalescent plasma, and I bet many of the people I see, there are 162 people listening right now, some of you have donated this. And this treatment has been used for over 100 years. And in fact, I don't know how many of you know who Balto is, but if I were showing slides, I would be showing you a beautiful picture of the statue of Balto, which is on 65th Street in Central Park. And Balto is famous for carrying antitoxin, which is a kind of something like a convalescent serum. It's a little bit different, but in the same general vein, from Anchorage to Nome to save the children of Nome from diphtheria. So that was I actually, for many years, thought it was at the turn of the last century, but it was actually in the early 30s. And the children of Nome were suffering from diphtheria, and the antitoxin saved them. And he carried that. People carried it, but he was the lead sled dog. And the Iditarod sled race is the memorial to that momentous trip. And that technology is a century-old technology. The notion is that if you have successfully recovered from an infectious disease, whether it be bacterial or viral, you have immunity in your blood. You have antibodies in your blood that are transferable to another person. And that's been proven time and time again. I have a whole lecture on it, but I shall not bore you with it, but take my word for it. And so literally what happens is, it used to be that the person would give whole blood. And during the Ebola epidemic in Africa, that was what was used, whole blood transfusions. But we don't want to take the whole blood from a person. We would rather just take the part we needed. So there are machines that filter out the cells and give them back to the donor. And just the plasma is removed, the clear straw-like fluid that contains the antibodies. The antibodies are what fights the infection. They are what's been mounted by the immune response. They're part of it. There's T cells, which are not transferred, and the antibodies, which are. And so somebody comes and they donate a unit of plasma. And a unit of plasma was good for 3 doses. So this is a technology that's limited by people. It's not scalable. And also is challenging because not everybody makes the same kind of immune response. Some people make a great immune response. Some people make a less good immune response. Some people really don't make any immune response at all. And there was no, by definition, because of the way it's done, there's no specific characterization to say, you've got a good immune response, we're going to take a lot of your plasma. So that worked, and it helped a lot of people. It probably saved a lot of lives. But it's a bit of an imperfect technology, both technically and scientifically. Monoclonal antibody therapy, which was invented by a colleague of mine named Michelle Nussenzweig at the Rockefeller, he was actually my boss for a while, is a brilliant, clever technique where you actually make a synthetic version of a very, very potent antibody, a highly neutralizing antibody. And it's been done for HIV, it's been done for Ebola, it's been done for Zika, it's been done for yellow fever. And I'm sure it's been done for many other illnesses. Those are just the ones that I know of in his lab. And the principle is that an individual, you screen people for a good response. So when Michelle and his clinical team, led by my very dear friend, Marina Kasky, did this at the Rockefeller in the height of the pandemic. And I can send the references to these papers because this has subsequently been published in Nature. Of 172 people who were screened, only 3 had really potent neutralizing antibodies. And everybody thought, oh, well, the people who got really sick and were in the ICU, they'd be the ones that had the really good, robust responses. And as so often happens, our prediction is wrong. You have to do the experiment. The people who had the best responses were people who had been mildly ill or who had been exposed to family members and hadn't gotten sick themselves. When you think about it, well, sure, that makes sense because their immune response protected them. So those people donate not their antibody, which is in the serum. They donate their B cells, which are the cells that manufacture the antibody. And those cells are interrogated, and the ones that actually make the really potent antibodies, because any normal immune response has myriad antibodies of varying potencies. So these are selected on a cell-by-cell basis. It's extremely tedious and hard work. And then they are made synthetically. And so this is the part also that's a huge advantage over the convalescent plasma treatment, because that you're getting from somebody else. It's screened to make sure it doesn't have other diseases. They do all sorts of things to it, but it's still a product from another human being. With the monoclonal antibody, the B cells are interrogated, the sequence is figured out, and then the sequence is used to synthetically make those antibodies at scale in a pharmaceutical factory. So there is nothing from the live person other than the information that is used in the actual drug. And so remember, I said they're very pure. So you can engineer, and my colleagues do, to pick, well, we want one to the receptor binding domain, we want one to this, we want one to that. And you kind of make a designer cocktail of what you have demonstrated in the lab you really need to neutralize the virus. And that's what my colleagues at Regeneron, which is, I have no interest in it and, financial, other than I just think it's a really cool company. George and Len, who are the founders, have done a brilliant job of really keeping high science while growing their company, and they've done a wonderful job. Johnson & Johnson also has a similar product, and there are many other that are farther back in development. These products are really good. They're currently being used to treat people who are very sick, and they're good at that, but they probably are almost more useful in being used to prevent disease. They could be given to healthcare workers, first responders, people in nursing homes. The problem with them is they're hard to make. They're not easy to make in its scale, because the scale we're talking about is hundreds of millions of doses. And so that there isn't going to be enough of them to use in all these ways. So right now, as you have to ethically, they're being used to treat people who are very sick. And, they're on their way to being approved. Both the Regeneron and the Johnson & Johnson one, and then there are many more coming down the pike. So those are antibody therapies that are passive. So you get as a patient, you are given an antibody, either from another human being or one that was made from the information from another human being. That's different from a vaccine. So that's called passive immunization or passive antibody therapy, and a vaccine is active immunization. And this is the really cool thing. Lenore?
About vaccines, there's a very common question about antibodies.
Sure.
Wondering how long they last.
Oh, that's a great question. And it depends on the antibody. Some of them, the half-life is how they're measured. Some of them last, only a week or two. There are ways to engineer them to, for those antibody aficionados, to modify the FC end of it. So that every antibody is shaped like a Y. Most antibodies, not every, there are exceptions, but for our purposes, every antibody is shaped like a Y. And this is the part that binds to the virus. And then the back of it, which is the back of the Y, is called the constant region. And that can be engineered to make them last much longer. So the HIV antibodies that my colleague Marina Kasky is developing, which have been licensed to Gilead, the first versions of them only lasted weeks, but they've been engineered in such a way that they last about 6 months now. So they can be engineered to last about 6 months. I think that's about as long as I'm aware that they can be engineered. But again, that's an engineering problem. And as you know, engineering progresses. And so they will be able to be engineered, I would guess, to last beyond that if they are not already. But again, that gets into a problem, right? So if they last 6 months, they have to be administered every 6 months, and that becomes a real challenge.
And for people who have had COVID and have their own antibodies, how long do they last?
So that's a very good question, and I don't have a very good answer. So I think the question that people really want to know is, how long does immunity last, right? So the antibodies are only one part of immunity, and frankly, for viruses, so the immune system has two arms: the humoral arm, which is the part of the antibodies that are made by B cell lymphocytes, and the cellular arm, which is composed of a variety of cells, but for our simple discussion tonight are the T cell lymphocytes. The T cells are much harder to assay. They require really complicated laboratory assays and fresh samples. So it's hard to know how long those responses last. People still have antibodies some people still have detectable antibodies at 7 or 8 months, now. Others don't. But it looks as though people probably, in the studies that have been done where they are seeing T cells, the T cell responses last much longer. But that's going to be a huge challenge because typically with other coronaviruses, the immunity only lasts, a year or two or even less.
Okay.
But one of the brilliant thing about the immune system is that it's characterized by memory. So you can have a memory to an organism, to a pathogen, and not have a detectable baseline level. The classic example of this is yellow fever. And, and again, I don't want to digress, but this is a beautiful experiment of nature. And a lot of what we learn are experiments of nature, things that happen that we can learn from, because obviously it's unethical to infect people, though that's another discussion. There's some talk about doing that in England, but let's right now say it is. And yellow fever, there was a yellow fever epidemic in Norfolk, Virginia. And I may have the dates wrong, but in the late, say 1890 something. And when it came back in the 30s, pretty much everybody who'd been alive in the 1890s was still immune. So, immunity is, when you get immunity, it can last a really good long time, even if you can't detect antibodies. So that's one of the great mysteries and one of the real challenges that we have to understand, both with natural infection and with the vaccine. And it's even more complicated because with other coronaviruses, so coronaviruses live in a family. There's the A part and the B part. The B part includes SARS-CoV-2, which is the virus we're talking about, SARS-CoV-1, which caused SARS in, I think, 2004, 2005 in China, and then MERS, which is the Middle Eastern Equine Respiratory Syndrome virus. Both SARS-CoV-1 and MERS are much more lethal, but much less contagious than SARS-CoV-1. But the A group cause common colds. So we have a lot of experience with them, both clinically and immunologically. And one of the tricky parts, and this is all data extrapolated from the common cold type viruses, you can be immune and not get sick, but you can still, as you probably know, the virus replicates in the mucosa of the nose and throat. So you can not get sick yourself, but still have virus replicating in your nose and throat, and therefore be capable of spreading it to other people. That's why even people who have antibodies need to wear masks. Probably it's unlikely they'll get infected themselves again, but it's not impossible that they could get enough infected that they could spread it to those people who aren't immune.
Do you want to talk about the vaccine now?
Sure, I'm delighted to talk about the vaccine. So the vaccine is active immunization. The term vaccine is actually, it's not trademarked, but it refers specifically to the smallpox vaccine, which was a virus called cowpox. It came from cows, hence vaca, hence vaccine. And it means the administration of, and it can be all different things. And the reason I'm mentioning that is in the 138 candidate vaccines, and in a vaccine that isn't licensed is called a candidate vaccine. There are all different modalities that are being tried to make a vaccine. Everything from RNA, which is the Moderna vaccine, to a chimp adenovirus, which has been engineered to express the genes of the virus, which is what the AstraZeneca Oxford vaccine is, and everything in between. So a vaccine is something that's introduced into your body, typically by injection, but it can be given intranasally as well, to fool your body into making an immune response that's going to be protective when you encounter the pathogen. So you get let's say the flu shot that I got last week, fools my body into thinking it's seen the strain of influenza that's currently circulating in Australia and South America. So hopefully when I encounter it in December, my body will, in a day or two, make a robust response and fight it off. One of the misconceptions that is a problem for some vaccines, but fortunately not for this one, and I think I discussed this with the class, Lenore, so, not to repeat myself for those of you who heard me on Monday, most people, if you ask, what is a vaccine made to prevent? What do you want to prevent with a vaccine? Most people would say infection. But the truth is, I am not aware of a vaccine that actually prevents infection. Most vaccines allow for 1 or 2 or sometimes even 3 rounds of replication of the virus when you encounter it. And it's that replication that boosts the immunity that you have from the vaccine. So for a virus like COVID, that's not a problem. But for viruses like HIV, for retroviruses, it's a huge problem. But what you want to prevent, what you want the vaccine to prevent is disease, ideally, but really what you want it to prevent is severe disease and death. So that brings me, there are a whole bunch of questions here. And one of the questions is, what percentage of effectiveness would you reasonably expect or hope for in an FDA-approved vaccine? And so that's a really important question. So typically, and all of these thresholds are set arbitrarily by a group of scientists, frankly, people like me, who think a lot about this and look at what the risks are and what the current state of the vaccines are. And they set thresholds, they have to be set before the vaccine trials start. They set thresholds, and then those are the thresholds that the vaccine developers have to live with. Typically, historically, for a vaccine to be licensed in the United States, it has to be more than 90% effective. Sometimes it's 93 or 95. The FDA has already said that they are setting the threshold for this vaccine at 50% efficacy, which is lower than any licensed vaccine of which I'm aware. The reason for that, it's not because it's not political. It's not, trying to get a vaccine by the election. It's because the modeling shows if you even have partial efficacy, you have a huge impact on the pandemic. So in the clinical trials that are ongoing, the immune response that is being seen, which is thought to be a correlate of protection, we don't have the protection data yet, that's still under wraps, is occurring in a much higher percentage of people than 50%. But the threshold that's already been, the preset threshold that's already been published by the FDA is 50%. So that would be what is referred to as a partially efficacious vaccine. There are 2 ways a vaccine can be partially efficacious. One is it works in, say, 50% of the people. And the other is that it doesn't protect completely. So it doesn't protect from disease, but it protects from severe disease and death. And often with the flu vaccine, that's what we see. I think it was 2 years ago, the flu vaccine was only a 23% match for the circulating strain. And though it probably didn't prevent as many people from getting sick as we would have liked, when the statistics were analyzed, it prevented people, and particularly vulnerable people, which for the influenza is older people over 80, and younger people, young children, from getting very sick and dying. So that's really what you need. I think most of us would be fine with, a week or 10 days of feeling unwell if we knew that we were going to be all right at the end and that there weren't going to be serious sequelae.
Someone just asked if the efficacy would be lower in older adults.
So that's a really important question. And our immune systems become what's called senescent, which is a lovely way to say that they peter out as we age. Our ability to make new immunity to a new antigen starts to decrease at the age of 2. So the thymus, which is the organ where all those T cells I told you about are made, it's the largest, its absolute size, not its relative size, but its absolute size in the human being is the largest when we're 2. So by the time you're my age, it's much harder to make a new immune response. However, there's no reason to think that this is going to be specifically harder than other viruses, but it is likely that people who are older, and particularly, over 80 or 85, they may not make as good a response as people who are younger. And this, needless to say, is a terrible concern because those people are most vulnerable to severe disease and death. many of you know that the flu shot, there is a special flu shot that is adjuvanted differently. It means that an adjuvant is something that's added to a vaccine to make it work better. It's an immune stimulatory molecule. So the current flu shot is, there's a different version with a stronger adjuvant for people over 65. That is currently not in the works for this vaccine, as far as I know. What I've heard people talking about is that people who are older will get, everybody for many of these vaccines will need a second dose, but may get a third dose as well. And that's what's being suggested with the flu shot. And that if you're older, especially if you got your vaccine early, so typically immunity from the flu shot only lasts about 6 months. So if you got your vaccine in August, peak flu is usually in February and March. You might want to talk to your doctor about getting effectively a booster shot in February and March. There is no additional risk to that. The negative of it, frankly, is the cost. And so fortunately for our community, I think that most people would opt for that second flu shot. Which brings me to another one of my community service announcements that I like to make. That is, please get your flu shot. Nobody likes it. Nobody likes needles. I don't like needles. I have, I just don't like them. I have one wonderful nurse who works with me, whose name is Arlene, who, whenever I need anything, she does it for me and lies me down and makes sure that I don't faint, because I've been known to do that, which is kind of embarrassing, but that's how it is. So I'm very empathetic to it. But this year is not the year to skip the flu shot. Even though it's an imperfect vaccine, our healthcare system is going to be overwhelmed with COVID. We don't want to overwhelm it with flu as well. People are talking about they're calling it the twindemic. Now, interestingly, people are also saying it's likely to be not such a bad flu season, because all of the conservative measures we're doing, wearing masks, washing our hands, keeping socially distant, will probably lessen the spread of influenza. And flu is carried from the southern hemisphere to us in two ways. One is by birds migrating, that's still happening, but the other is people coming from Australia and South America, and there's much less of that. So it's very important to get your flu shot, but in my ever optimistic way, I'm hoping for not too bad of a flu season.
And what can the scientific community do to assuage public fears about the safety of the vaccine?
That's a huge question, Lenore. And, my area of expertise is really the science, but every time I talk publicly about this vaccine or any other, it comes up. As everybody knows, there are a community of what are called the anti-vaxxers who have their worries, and they have their worries about vaccines that have been developed, in the fullness of time, as I like to say. This vaccine has been moved forward at warp speed. And so typically vaccines take a decade to develop. As far as I know, the fastest vaccine that's been developed is the measles vaccine, which was developed by Maurice Hillman, who actually isolated the strain that was used, the virus that was used to make the vaccine, from his daughter's throat. His daughter was sick and he cultured her throat and he went running to the lab. And it took 4 years for that vaccine to be developed. So that's kind of the record. So the fact that this has been developed so rapidly is a testimony to a tremendous amount of work and a tremendous amount of money spent. My colleagues, and frankly me, and people who help us with communications are going to have to help with the messaging, because even people who aren't anti-vaxxers are questioning it. One of the things that I think is very helpful to understand is where this has been rushed. So one of the things that has been done that I think was actually brilliant to do was this vaccine has been made at risk. So typically a vaccine or a drug starts out in the lab, it moves into early phase studies. So phase 1, which is safety only, start with a very low dose. So you start with the tiniest dose you can after you've shown it to be safe in animals. And then you move up to a larger dose, and then you move up to a dose that you think may actually work. And you do that in 12, 14, 24, depending on a variety of things, numbers like that of people. And then if that works, you move to a phase 2 study, which phase 2 for drugs is dose finding, but for vaccines is immunogenicity. So that's where you're interrogating the blood to find out if people are making antibodies, they're making neutralizing antibodies, if their T cells are responding. And that's where, when I'm saying the data is very promising, it's from the phase 2 study data that's been published. It's in the New England Journal. I can send you reams of articles, or you can just look them up yourself. It's quite comforting. And then only with a phase 3 study do you find out if a vaccine works. So in a phase 3 study, you enroll a lot of people, tens of thousands of people, and you randomly assign them either to get the vaccine or get a placebo. And a placebo can be basically salt water or a vaccine that's not relevant. In a trial that I was involved in Thailand for an HIV vaccine, the placebo was a rabies vaccine, because we wanted to give people a benefit. In a rabies vaccine, there's a benefit. And then you follow those people. You wait to see if they get sick. And that's why nobody can tell you when the trial is going to be over, because you don't know how many events are going to accrue. That's the term of art, which in this case means how many people are going to get COVID. The people come in and they get tested. I don't know. The protocols are actually available online. You can read them. I don't know if they're getting tested every week or every 2 weeks, but it would be something like that. And if all of the infections fall in the placebo group, or almost all the infections fall in the placebo group, your vaccine worked. If they fall equally in the two groups, it didn't. If they fall more in the group of people who get the vaccine, that's a whole other talk, but that's not going to happen here. There's no indication of that. So you wait for those results, and then if a company is doing this, because companies are in the business of making money for their shareholders, then and only then do they go ahead and manufacture the vaccine. And the manufacturing of a vaccine at scale is, to say it's non-trivial is an understatement. It's a huge endeavor. It's expensive, it's hard, it can go wrong 6 ways till Sunday. And so, like with the polio vaccine, which is, polio was a scourge, not exactly like this, but for those people who lived through the polio epidemic, they'll tell you, people lived in fear, it was dreadful, and it was particularly dreadful because it affected young people and children. So children were dying and getting paralyzed. And the thought of that just makes my, the hair on the back of my neck go up. The Salk trial ended, and it took 1.5 years to make the vaccine. There were no plans to make the vaccine at the time of the end of the trial. So polio, unlike this, is a summer virus. There was a whole other season. There were a whole group of children who got sick, died, and got paralyzed, even though a vaccine was known to work, because it just hadn't been made and distributed. So the government, under the Operation Warp Speed, has made and vialed, and putting the stuff in vials is a whole other important and hard and challenging activity. And for a while in the late summer and early fall, there was a shortage of the glass vials, and they had to do all sorts of things to get the right amount of vials and stoppers. All of these mundane things, one of them goes wrong and everything stops. So the vaccine, when people ask, is there going to be a vaccine? My honest answer is there is a vaccine. There are actually 2 in vials waiting for the results of the trial. And that's never been done before. So that's done at risk. We are paying for it. The taxpayers are paying for it, and that's billions of dollars. And that was done, I think, very wisely. Because how would we feel if in December we found out, yes, the Moderna vaccine works, but it's going to be another 1.5 years before it's going to be available? I don't think any of us would be very happy about that. The risk is, if it doesn't work, you're left with billions of dollars of vials filled with something that you don't want. But our government has taken that risk on. So I think understanding where the time was gained. So it wasn't that the safety studies weren't done. It wasn't that the safety data hasn't been gathered. It's that the manufacturing process was compressed in this way. I think understanding that is helpful in reassuring people. It's helpful to me. I think the FDA, I will tell you, the FDA is not a behemoth. The FDA is a group of very dedicated, very smart, hardworking, thoughtful regulatory scientists. And they have been clear about the fact that they are going to apply the pre-stated requirements to this vaccine. There was an external advisory committee meeting today. In fact, I got the link to it, and I'm happy to share that if anybody wants to watch, the discussions around this, what is going to be required. And part of what's going to be required is how long are we going to wait to see safety data, right? So you get the efficacy signal and you only have safety data for 2 months after the vaccine was administered. Is that enough safety data? Do we need to wait another 2 months? That's one of the discussions that's going on now. So there's the issue of efficacy. Will the thing work? And will it work in the elderly, say? One of the problems is it hasn't yet been tested in children. It has been tested in older people. Moderna enrolled people 65 to 85, I think. My mom actually tried to enroll, which I was so proud of her for doing. So it has been tested in that very important vulnerable population. Typically, all drugs are tested first in adults, with rare exceptions, unless they're only for children, and then when demonstrated to be safer, then tested in children. So I don't know where the trials for children stand, but children won't get it until that data has been collected, certainly.
Someone asked also before on that subject, do you know if people with antibodies are also part of the trial?
Yes, they are.
Okay.
They are, because it's very important. But, one of the things that's really interesting about this virus, and as horrible as all this is, and it is so horrible, I actually feel badly because, I didn't say at the beginning, my sympathy and our prayers are with all of people in our community and other communities who are ill, who have lost family members. There are what now they're being called the long haulers, people who got sick and aren't getting better so fast. And I can only imagine how dreadful that must be. So, this virus is wreaking havoc everywhere, let alone the economic implications of it. And so, I'm sorry, Lenore, I just completely lost the question. Can you remind me of what it is?
I was actually going to ask about the timing, but we were talking about people with antibodies.
Right. So the big question, thank you. One of the big things that's remarkable is, unlike, say, Ebola, where virtually everybody without antibody therapy or some of the drugs now, 100% of people who get Ebola die. I mean, virtually everybody. The type A coronaviruses, the colds, pretty much everybody gets a cold or nothing. They don't get, there's no serious sequelae. This virus has it's called polymorphous, a huge variety of outcomes. 40% of people supposedly don't get sick at all. Most people get what's called mild disease, which is a flu-like syndrome for 10 days to 2 weeks. And then there are some people who get pneumonia, all of the things. There are people who get neurologic involvement, there are people who get cardiac involvement, and then of course there are people who sadly who die. There is certainly a genetic component, and a colleague of mine named Jean-Laurent Casanova with a team of hundreds of people from around the world has discovered that in the outliers, people who are young and have gotten very sick or died, and people who are old, like 105 and survived, they have polymorphisms in the interferon pathways, which one would predict, but he's demonstrated it. So there are underlying genetic conditions that make one predisposed, in addition to all of the clinical conditions that we've all heard about. Age, male sex, having a variety of clinical conditions. But one of the other things, and this was a long way to get to your question, is what may protect some people is they had a bunch of common colds, and there's cross-reaction between the coronavirus antibodies from those group A coronaviruses and this current one. So it's very important to understand how the vaccines will perform in people who have antibodies, because a lot of people will have them. There's no reason to think they won't be safe, because we have antibodies to flu all the time, right? We've had the flu, we've gotten immunized to the flu, we get immunized every year to the flu, we get immunized again to the flu. So it will likely be safe, but it is being looked at specifically for sure.
Do you think we'll need to be immunized every year for COVID?
So that's an important question, and I saw it in the chat, and people ask all the time. So the reason that we need to be immunized every year for the flu, so the flu vaccine, and I say this with a full measure of respect to my flu colleagues, it's a terrible vaccine. It's a terrible vaccine for a variety of reasons, and I'm not going to go into it. But the fact that we need to be vaccinated every year is really bad. And part of it has to do with the quality of the vaccine, but part of it has to do with the fact that influenza mutates tremendously dramatically. And it undergoes two types of mutation, one called antigenic drift, which is kind of the general way viruses mutate. A random error is made and it's promulgated, but then they undergo something called antigenic shift. And so that's where the hemagglutinin and the neuraminidase, which are the major proteins, like a whole cassette switches out. So that's why you hear H1N1, that's hemagglutinin 1, neuraminidase 1. And so that's the one that circulated this year. And then next year we have H2N3. And we haven't seen N3 in 50 years. So that's going to be bad because people don't have immunity. Fortunately, we don't have that with COVID. COVID is a pretty stable virus. It's a pretty ordinary virus in that way. So I think eventually we'll probably have a good, more what's called durable vaccine. But the current vaccines are almost certainly not going to be the ultimate vaccine. And this also happened with polio. The first vaccine that was developed, everybody got, and then a year and a half later, Salk developed a vaccine that was made by killing the virus with formaldehyde. It's called a whole killed virus, a whole kill vaccine. And a year and a half later, Albert Sabin developed the live attenuated vaccine, which is a polio virus that had been grown in the lab over and over and over again. So it was weakened and conferred protection and not disease to most people. And those are the polio drops that most of us adults got. So people got one vaccine and then they got the next vaccine. And that's fine. That's called a prime boost strategy. And that's done intentionally with an HIV that's done. And so my guess is that we know there are many vaccines and that many of us will get one and then get the other. Will it be possible eventually to have a durable vaccine? I certainly hope so. I don't think these early vaccine candidates are going to be that vaccine. I think we're all going to have to be vaccinated. Certainly many of them require 2 doses, and it's clear they require 2 doses. So that's a challenge. We're going to have to, like the HPV vaccine, we're going to have to be sure to go back and get that second shot. But I think that there will probably be other vaccines down the road that will be more efficacious than this first group.
And do you want to share your predictions on timing and who will it be available to first?
Absolutely. But what I want to say is, in my mind, and I have this sort of gut standard that I use, as we've discussed and Kenny so graciously mentioned. I am known in many circles as Jacob's mom, which I am proud of every time I hear it. I am also Josh, Zach, Daniel, and Joseph's mom. And I am fortunate, and I have wonderful stepchildren who I dearly love too. And we have 3 granddaughters, Noah, Shira, and Arya. And so I have this wonderful, beloved group of young people. And so in my head, I have what would I do myself? And I'm willing to take fairly more risk with myself. And what would I give to my kids? And so I wouldn't recommend to anybody else something that I wouldn't give to my children. And I think that nothing is perfect and everything has risk. And the risk of getting COVID without a vaccine, to me, is a pretty scary thing. So I'm much more focused on the safety rather than the efficacy. So for instance, the Moderna vaccine, which is the RNA vaccine, I'm really optimistic about that. Not so much in terms of efficacy. I think the efficacy will be good. It might not be great, but I think it will be safe. It's not, it doesn't overstimulate the immune system. I'm very optimistic about that. There are most of the vaccines I'm very optimistic about in terms of safety. And how efficacious they'll be, we'll see. But that will be okay, because as I said, the modeling shows that even if they're 50% efficacious, and I think they'll be more than that, they will really, really change the course of the pandemic. And that's obviously what we all need for all the reasons we know. So you asked me about timing. And I think I mentioned, and this is one of the questions. So, we talked about this with your seniors. Understanding the vaccine development process and how it proceeds is really helpful in understanding why we don't have answers. It's not that that anybody's obfuscating or hiding. It's because these are what are called event-driven trials. And you have to wait till the events accrue. So actually, the fact that things got better in some places, like here in New York in the summer, slowed them down. Now, of course, things are going up again, so they may end faster. The other thing that I wanted to mention is people, the trials get paused for safety. and people worry about that. Well, in my mind, that's the indication that the vaccine developers are doing exactly the right thing. Because what happens is you have these two groups of people, one group that got the vaccine, one group that got the placebo, which you know to be inert. And things happen to people, right? Things happen all the time. People get sick, especially if you're enrolling people 65 to 85. People get sick, die, they get hit by buses, they break their ankles. Anything like that that happens has to be reported, and has to be reported to, there's an outside safety group called the DSMB, or the Data Safety Monitoring Board, and has to be reported to the Data Safety Monitoring Board. So for instance, somebody breaks their ankle and gets admitted to the hospital for a repair. That gets reported to the Data Safety Monitoring Board, and they have to review it. Well, is that related to the vaccine, or is it not related to the vaccine? One would think, well, it's probably not. But there have been cases with certain kinds of drugs, not with vaccines, where they've had neurologic toxicity, and there have been increased number of falls and then increased number of broken bones in the people who got the drug. And so therefore, DSMBs are very conservative about investigating what happens. That's their job. And that's what we want them to do. So a pause in a vaccine trial is both normal and an indication of careful conduct of the trial. So those things get announced in the paper and everybody gets very worried. One of my favorite personal stories about that is I was doing a vaccine trial, and we had this poor woman who had every symptom in the book. She had pelvic pain, headaches, on and on. And I kept spending time trying to figure out how this vaccine, which frankly wasn't very immunogenic, was causing all of these things. And then at the end, when we broke the blind, she had gotten placebo. Thank God she got placebo, because it was a very small trial. And if somebody had had all those complications, it would have really had a negative effect on the vaccine. So we don't even know if the people who got sick received the vaccine or got placebo. And it's appropriate that we not know that.
Someone asked, since the vaccines won't be close to 100% effective, and there are people who will opt out of having the vaccine, what will life be like after we're vaccinated? Will we still have to wear masks, social distance?
I don't know. I wish I knew the answer. I ask myself all the time. This too shall pass. It will end. We will go back to the theater. We will go back to shul. We will, I'll get to have my break fast again, God willing. But I don't think things are going to be the same for a while. But I really think they're going to get better in the spring. I think we're going to have a hard, late fall, early winter. And then I think even if the vaccines aren't perfect, even if we still have to wear masks, I think we're going to be able to move around with more comfort. I think the rates are going to go down. And, the thing that drives the epidemic the most is the rate of infection in your community. So in the spring, when we were walking around and 20% of the people around us were infected, it was super important that we had a mask on, because there, 1 in 5 chance that the person you were walking by had the virus. Now, I don't know if that's actually right, but that's what the testing was. In the middle of the summer, it was so low, that it was 1 in many hundreds. So the chances were most of the people you encountered were uninfected. And so all of your protective measures didn't matter so much because there was no virus there. So as these things come on board and they drive down infection, we'll be safer. And I think it's going to take time. I think it's going to be slow, slower than I had thought perhaps in the spring. But things will go back. And I think that we have to have faith that they will. One of the other things that I haven't spoken about today, and I'm keeping an eye on the time, is testing. So as all the therapies, the drugs, the immunotherapy, and the vaccines are moving along, so is testing. So if you think about it, if you could, instead of filling out I forget which health app, what the name of the health app for Ramaz. Magnus health app, because I have that one, I have the one for work, all the health app, that I have to fill out. And for work, I have to fill it out 3 hours ahead of time. So I have to remember, and it has to be whatever. You fill that health app out. Instead of doing that, if you could spit on a strip, take a time-stamped picture of that strip, and submit that and show that you weren't infected, and know that everybody else who was coming to your work or your school wasn't infected, that would change everything. So the testing is moving forward too. Granted, more slowly, frankly, than I'd hoped. like so many things, it's turned out to be more challenging, but the testing is moving forward too. So that in some ways is going to facilitate opening perhaps at least as much as these other measures. I just heard Governor Cuomo say that, there's all, basically every state in the nation except Connecticut, New Jersey, and Pennsylvania have to quarantine when they come here. And Connecticut, New Jersey, and Pennsylvania meet the requirements. It's just that from a logistic point of view, it's not possible. And he's saying now they're looking at if there are testing algorithms that can be put in place because so much more testing is available. And that's a hugely good thing. And so, as you know, Lenore, I'm an anxious person. And when I worry about what it was like in the spring, things that are different now are, we know a lot more about the virus. So if people get sick, we know how to take care of them better. The shortage of PPE, shortage of masks, and frankly, the inconsistent messages about wearing masks are completely changed. And in addition to that, testing. Remember you couldn't get a test, and then if you could get it, you didn't know if it was right or not, and it took a week or 2 weeks to get it back, and at that point it was useless. That's just not the case now. So those things are already improved and I think are making our lives better currently.
I'm just keeping an eye on the clock. It's almost 9 o'clock, and I know you want to get to your core 4.
Yeah, I got to get to my core 4, and I think I have all of my props here. So the core 4, which I think it's actually core 5 because there's another one, but the core 4, and that's not my my terminology, but it's public health terminology. So all of these things that I've talked about are on the horizon. They're on the close horizon, but we don't have them now. What do we have now that works? The single thing that works the best is this, the mask. I can't tell you, and I don't want to review the data, and it upsets me so much that this wasn't communicated clearly back in February. We would have a whole different epidemic if that were the case. This kind of mask, or a fabric mask, or even surgical masks, protect you and they protect other people. So they keep you from getting the virus in, and they keep you, if you have the virus, from sharing it with others. And they do it really, really well. There is also some thought, and I'm probably going to go a minute or two over, but I want to go over these points because these are really useful today. There is a lot of data to suggest there's what's called a dose effect. And that means if you get a bigger dose of the virus, you're more likely to get sick. There are some viruses that have that and some don't. Chickenpox is famous for that. Many other viruses don't have that, but this virus very clearly does. And that means that if you have, you get somebody sneezes in your face, you're much more likely to get seriously ill. And the masks, even if you get a little bit of virus, you're much less likely to get seriously sick. So I personally prefer this kind of mask. My husband, my beloved husband, Jacob's dad, Ellie, likes the ones that, do I have it here? No, that go over the head, not over the ears. But everybody has their own favorite kind of mask. I find having a chain makes it much handier, so I don't miss, so I know where it is. The fabric masks work really well. The things that don't work well and to avoid, I've kept this mask. I threw out all the other ones. I loved this one because I bought it in the summer and it was black gingham, which I thought was really kind of cool. But this valve is a disaster because that valve lets you blow out virus. So you are being a bad citizen by wearing a mask like that. I see people on the street with them, and I never correct people. I don't want to get into it, but I want to say to them, please get a mask without a valve. I don't think they should even be allowed to sell these now. And I've only kept this one. I threw all the other ones out for my little demonstrations. In addition to a mask, glasses actually help. There was a lovely little study done in China in a province, and I forget which one, where 30% or 40% of the population wore glasses, but only 6% of the people who were hospitalized with COVID wore glasses. And what that suggests is probably they provide some level of protection for your eyes. But whether you wear glasses or not, this thing, the face protector, is great. And this is what my colleagues in the hospital wear when they're taking care of patients. And so if you're at high risk, or if you are doing something that's risky, or if you're going someplace
where you think people are going to be infected. This on top of this, not instead. Again, sometimes I see people on the street wearing this instead, and I want to say, don't wear that, but I don't. Really, really afford tremendous protection. Now, obviously, they're uncomfortable, they're unpleasant, they have all kinds of social ramifications. You can't hear people, you can't see their faces. Everybody's exhausted, but we have to keep it up. So the core four are wearing your mask, hard but feasible, but you have to wear it every time. And none of this with the nose out, you see people on the chin, none of that. That doesn't work. Find a mask that's comfortable for you and wear it. Wearing your mask, wash your hands, and that's washing your hands for 20 seconds, and that means 2 courses of happy birthday, fronts and backs. It's a lot of hand washing. Fortunately, it doesn't turn out that surfaces are as much of a problem as we thought they were at the beginning. In the beginning, if you listen to some of my old ones of these, I talked about my neurotic disinfection of my groceries. I am no longer afraid of my groceries. I am no longer disinfecting my groceries. But in the core four, there's the hand washing, the masks, staying socially distant. So staying socially distant, 6 feet is what's recommended. Outside, that works great. Inside, it's not enough. There have been several cases of people indoor dining greater than 6 feet apart. And obviously when you dine, you have to take off your mask, where people have gotten infected. So that's something that really worries me a lot for the community. And when we talk about doing things in the shul, like we can't be, my favorite thing is to eat with my family. My greatest pleasure is to have everybody for dinner, for Shabbat, for holidays, for Hagim, for any occasion, pizza night on Mondays. My boys, my mother comes. We all, it was like this lovely ritual. We can't do it. Indoor dining is terrible. I wish it weren't the case. Outdoor dining is better. Outdoor seems to really confer a level of protection. Opening your windows helps a lot. Artificial ventilation isn't good because it's blowing the virus around. And there are a bunch of videos which I can send where you see people sneezing and then you see the particles floating in the air. So being outside, and that's what's really going to get hard. Sukkot was lovely. We were able to be outside and distanced with our masks on. That I get tearful thinking about Thanksgiving, about not being able to, have dinner at my mother's house. We can't do it. The data is showing that people are not getting infected in their places of work. Thank God they're not getting infected in schools. People are getting infected in small groups. We're all so tired of this. And just because we're tired, it doesn't mean the pandemic is over. It will be over, but it isn't over yet. And so we really have to double down. And Zoom is horrible. Jacob says Zoom school is like school with none of the fun. And I tell him Zoom work is the same for me. It's really hard, but we've got to do it. So good to go back to my core four: masks, hand washing, social distancing. So 6 feet outside is good, but inside, don't take your mask off if you're, within 6 feet. The social distance isn't enough. And then the last one is don't go out if you're sick. So, in past years, I'm the mother who's like, you look fine to me. Let's go to school. We'll see how you feel. We can't do that this year. If you are unwell, if you feel unwell, if your child feels unwell, stay home. And that means really stay home. Don't go in the elevator. Don't go anywhere. Stay home and get tested. The other last little bit I would just remind people of is elevators are terrible for ventilation. My husband is in real estate and he constantly reminds me there's no ventilation in elevators and the virus lingers in the air. And there is this inclination because you're alone in a closed space you can take your mask off. Don't. Same with public restrooms. If you have to use a restroom, the surfaces probably aren't the danger we thought they were, but what's lingering in the air, be quick, keep your mask on, wash your hands well, use a paper towel to touch the surfaces. So those things are not cool and sexy. They're not high science. They're hard to do every time. And unfortunately, you have to do them every time to make them work. But the really good news is they work. And we now know enough about how the virus is spread to know that they really can protect us and our children. So.
Thank you. Thank you, Sarah,
Thank you.
for another informative, amazingly informative evening and giving us a lot of wonderful advice. Lenore, thank you for moderating and for helping us to think about many things that we need to be thoughtful on, and both of you for teaching, educating our children in all of these things. And it is essential that as we go forward, we will continue to understand and to learn as much as we possibly can about this topic. So thank you.